In this activity, you will examine the internal anatomy of a Eudicot leaf using a prepared cross-section. While Eudicot leaves contain the same major tissue systems as monocots, epidermis, mesophyll, and vascular tissue, the organization of the mesophyll is distinctly layered, reflecting specialization for light capture and gas exchange. In Eudicot leaves, the mesophyll is divided into two distinct regions. The Palisade mesophyll, located just beneath the upper epidermis, consists of tightly packed, elongated cells rich in chloroplasts and specialized for photosynthesis. Below this is the Spongy Mesophyll, composed of loosely arranged cells with large air spaces. These air spaces allow carbon dioxide and oxygen to diffuse easily between the stomata and the photosynthetic tissues. This layered organization contrasts with the more uniform mesophyll of monocot leaves and reflects a division of labor within the leaf. Before observing the slide, answer this pre-lab question. Why are Palisade mesophyll and Spongy mesophyll arranged differently in a Eudicot leaf, and what advantage does this provide? Think about where light enters the leaf, where most photosynthesis occurs, and how gases must move in and out of the tissue. Record your prediction in your lab notebook before continuing. Before you make your labeled drawing, let's review each structure so you know exactly what you are looking for in the microscope and what you will be labeling. The upper epidermis is the outermost layer on the top surface of the leaf. It consists of tightly packed cells that protect the leaf and help reduce water loss. Just above it is the cuticle, a thin waxy coating that further limits evaporation and protects the leaf from physical damage. Directly beneath the upper epidermis is the Palisade mesophyll. These cells are long, column-shaped, and tightly packed. They contain many chloroplasts and are the primary site of photosynthesis. Because light enters the leaf from the upper surface, this layer is positioned to capture as much light as possible. Below the Palisade layer is the Spongy mesophyll. These cells are more loosely arranged and irregular in shape, with large air spaces between them. These spaces allow carbon dioxide and oxygen to diffuse easily through the leaf, making this layer important for gas exchange. The lower epidermis forms the bottom surface of the leaf. It is similar in structure to the upper epidermis, but usually contains more stomata, which regulate gas exchange and water loss. Within the mesophyll, you will see the vascular bundles or veins. In this view, this is the large midvein of the leaf. You will not see other veins in the cross-section of the Eudicot since they have net-like veins, so instead of being parallel with the midvein, they are branched out in different angles. This is depicted in the right graphic. Inside each vein, the xylem is typically located toward the upper side and consists of larger, thick-walled cells that transport water and minerals. The phloem is usually found toward the lower side and consists of smaller, thin-walled cells that transport sugars produced by photosynthesis. Surrounding the xylem and phloem is the bundle sheath. That encloses the vascular tissue, although it is hard to clearly see that in this view. These cells help regulate movement of materials into and out of the vein and provide structural support. As you observe your slide, locate each of these structures and use their position, shape, and cell arrangement to guide your labeled drawing. Now create a detailed labeled drawing of the Eudicot leaf cross-section in your lab notebook. Label the upper epidermis, cuticle, Palisade and Spongy mesophyll, lower epidermis, xylem, and phloem. Pause the video while you complete your drawing. Complete the following compare and contrast questions. First, compare the mesophyll of monocot leaves with that of Eudicot leaves. Think about layering versus uniformity in the mesophyll. Second, how are the veins of Eudicot leaves similar to those of monocots? How are they different? Focus on xylem and phloem position, but also overall vein arrangement. Lastly, how can you explain the differences between Palisade and Spongy mesophyll in terms of their structure and function? Connect cell shape, packing, and airspace with the processes of photosynthesis and gas exchange. Write your responses in your lab notebook. Eudicot leaves show internal specialization through the separation of mesophyll into Palisade and Spongy layers. This arrangement allows efficient light capture, gas diffusion, and transport, demonstrating once again how plant anatomy reflects functional demand.